Ladies and gentlemen, it's a great pleasure for me to be here today and to talk about the topic from super resolution microscopy to zebrafish, investigating FSGS and other glomerulopathies. My name is Nicole Endlich, and I'm the Managing Director of the Department of Anatomy and Cell Biology at the University of Medicine in Greifswald, as well as the CEO of the startup of the University and University of Medicine named Nipoca. Some words about my background. I'm now more than 25 years in the field of the podocyte research, and here in the northern part of Germany, I'm working since 20 years on this topic. Now, as you know very well, chronic kidney disease is a global burden. And more than 10% of the people worldwide are affected by this disease. There's also an increasing death rate, and it is expected in the year 2014 that it's on the position 5 of the mortality rates. Causes and risk factors are different. So it could be diabetes, high blood pressure, obesity. You can have a genetic background, which is responsible for the disease, circulating factors, as well as the age. So my lab and startup is focused on podocyte-related disease, and you will see images and information from both sides, from my lab as well from Nipoca. The first part will show you some details about super resolution and podocytes. And podocytes are really fascinating cell type in the kidney, in the special filtration unit of the kidney, the glomeruli. And as you can see here with the scanning electron microscopic image, you have a specific cell type sitting at the outer aspect of the capillaries, and they have major processes and tiny so-called foot processes. In more than 75% of the chronic kidney disease patients, these cell types, the podocytes, are affected. If you look here more in detail, you see the regular interaction between the neighboring foot processes and the formation of a so-called filtration slit in between. This morphology is highly relevant for the size selectivity of the kidney filtration and function. Therefore, any changes, as you can see here, a broadening of the foot processes will result in a change of the filtration selectivity. In many glomerulopathies, the morphology of these podocyte foot processes are affected to various degrees. Now, in the past, there was only the possibility to study the podocyte foot process morphology by the electron microscopy technology, since the size of the podocyte foot processes is below the optical resolution of a normal light microscope. The background of this is the Ernst-Abbe law. And this law described that the resolution is dependent on the wavelength which is used for the illumination. And therefore, the optical resolution of a light microscope is limited to 200 nanometer. As you can see here, electron microscope is the only way to visualize foot processes and the filtration slit in between. However, there are several limitations of this technology. It's really a highly sophisticated technique and time consuming. You have only 1 to 3 glomeruli with a thickness of 50 to 90 nanometer for your analysis. There's a specific embedding and fixation necessary, and the analysis is always subjective with limited reproducibility. I would like to show you an example. You see here in this slide on the left-hand side, a healthy situation. It's a part of the glomerulus. You see the GBM, the glomerular basement membranes, the endothelial cell. This is the lumen of the capillary. At the outer aspect, you see nicely formed foot processes. This is easy. This is a healthy situation. On the right-hand side, you see the contrast. You see a broadening of these foot processes, the GBM again, and here's the endothelial cells, the capillary lumen. And this is clear, this is a diseased situation. However, how is the situation in between? You have areas with healthy podocyte food processes, you have with phased food processes, as well as something in between. The analysis of the effacement is also not easy and always subjective and require an expert due to the digestion angle bias, and therefore you need a lot of experience. There's also the problem, as I mentioned before, that there is no measurement, no value for the changes of the podocyte food processes. It's like if you would like to know something about your weight and you only see too much instead of an exact measurement. And now it is possible with a new technology called super resolution microscopy to change this and to present a value for each kidney and for each glomerulus and patient. Furthermore, I have had the question in the past, are really 1 to 3 glomeruli from 25,000 in mice for research and 1 million in human with a thickness of only 80 nanometer really representative? And therefore I asked myself, is there any possibility to make it a measurement fast, exact, unbiased, and reproducible? Yes, it is. There's a new type of light microscopic technology that overcomes the physical limitations I have shown you before. And this is a so-called super resolution microscopy. And I will show later something about a specific technique. So it's called SIM. But some words about the people behind this development of this fantastic technology. You see here the people who developed this technology, Hell, Betzig, and Murner, and they have got the Nobel Prize for this. And there are different technologies altogether, the super resolution microscopic technology. And here we have SIM, stands for structured illumination microscopy, STAD for stimulated emission depletion microscopy, STORM, and PALM. However, for us, it's working if we are using SIM, the structured illumination microscopy, to visualize food processes as well as the slit membrane, and I will show it to you a little bit later. Now, the consequences of this development is that it accelerates research and drug discovery, and it will revolutionize transplantation analysis and personalized medicine. But first of all, I would like to show you how it works. So you have again here this image from the nicely formed food processes of a podocyte here indigitating with the neighboring food processes. And in between, as I mentioned before, there is a filtration slit composed by the protein, transmembrane protein called nephrin. This is not visible in this illustration. Now, what is the super resolution microscope doing? And this, as I mentioned before, is a new light microscopic technique that overcomes the physical limitation given by Ernst Abbe. And so we have now an optical resolution instead of 200 nanometer of 100 nanometer. And this is enough to visualize the food processes as well as the filtration barrier in between. You see here an example. This is one part of a glomerulus stained, of the kidney stained with an antibody against a filtration slit like nephrin or podocin. And here, if you take it with a wide field, the image, you see it only in a blurry way. However, if you have a super resolution image, then you can clearly see these green lines. These are important lines. This is a filtration slit. And the black areas in between, these are the unstained podocyte food processes. So we named this technology of the visualization and later the measuring of the changes of the podocyte food processes in abbreviation PEMP, and it stands for Podocyte Exact Morphology Measurement Procedure. Now, what we are doing is we take a normal standard histological slide formalin fixed and paraffin embedded section. As you can see here, no special treatment, a normal slide, as you have it very often in the pathology department or in your research lab. Then we make a double staining. One antibody is directed against the filtration slit, like nephrin and podosine proteins, and one against protein specific for podocyte food processes, as synaptopodin or integrin alpha-3. After sustaining, so we have, here it's an animal example, a cross-section of the kidney, and all these colored areas are the filtration units, the glomeruli, selected by an AI-based segmentation tool. So here you can see it in real. Here you see single glomeruli and the AI-based segmentation. And on the right-hand side, you see in a higher magnification. Now we make in the next step, the image acquisition with a three-dimensional structured illumination microscopic technology, meaning we make not only one image, we make a so-called optical sections. We are going through the tissue of the 3 micron with a laser beam. And we take also information of these optical sections. And I have to move here my image a little bit. Okay, then you see here, this is the visualization of the filtration slit of one glomerulus. And this is in a color-coded way after the images of these optical sections were put together as a so-called maximum intensity projection in a privation MIP. And the color indicates where these filtration slits are located, on the upper side or on the bottom side of your slide. And you can now focus in, for example, in area A, you see something has changed with the filtration slit, and in B, you see a healthy situation. We have tightly packed filtration slits together. Now, the next step is that we make a measurement, and the measurement is you take the length of the filtration slit. Here you have seen in the green color before, and you divide this through the area where the podocyte food processes are located. And this is a so-called filtration slit density. And the value is here as an example, 4.1 micrometer per square micrometer. What is background of this FSD? I would like to show you this here in this example. If the FSD value is reduced, then you have an effacement. And you can explain it here with this cartoon. If you have a healthy situation, these are interdigitating podocyte food processes, then you have a very long filtration slit in between. In contrast, if you have an effacement, then you have a reduced filtration slit and therefore also a reduced FSD value. So here's an example. And on the left-hand side and here on the top, you see again in green, this is a filtration slit, nephrin or podosine. In this example, it's podosine. In the middle, you see the staining. It's a double staining, as I mentioned before, for the food processes done by synaptopodin. And on the right-hand side, you have the merge of the both images. And you see in magenta, the food processes, and in green, the filtration slit in between. I think it's clearly visible, this disease situation. You have a reduction of the filtration slit, and you see here, this is a food process, here on the right-hand side, in combination with synaptopodin, which is broadened in comparison to this or that food process. Now, how it works on human biopsies. I would like to show you this example here. You see a biopsy, and all the glomeruli are stained, double stained. And now we zoom in and we look now for an exact situation in this super resolution image. This is one glomerulus stained again with podosine and synaptopodin. And now we can zoom in at every region we like and to study the morphology of the food processes. This is a filtration slit, and in between here, the magenta indicates the food processes. This region looks pretty nice. However, it could be that in one glomerulus, you have different areas with the food processes. Here is an example in a higher magnification here on the top side. You see the filtration slit, and it's tightly packed, and the food processes are tiny. In contrast to the situation here, shown on the left side here and on the bottom here, you see that something is wrong, and this is a severely affected podocyte food processes as an example. Again, this is the situation if you compare it directly. So another example showing membranous nephropathy. You see a lot of podocytes are still attached. This is a glomerulus, and you only see stained the filtration slit in this area and the podocyte here with synaptopodin indicated. And you see again, there's a change of the filtration slit length. Now we have at the beginning used biopsies from patients suffering from minimal change disease. And at the beginning, we have taken 13 patient biopsies, and now we have used several hundreds. But it illustrates it very clearly. Patient 1 has a different FSD than patient 2. You see it here, FSD measurement 1 to 4. And the healthy group is taken together in this green circled area. Again, high FSD value means healthy, lower FSD values means diseased, and if you have a very low FSD, then the glomerulus is highly damaged. You can use these measurement and observation for all kinds of glomerulopathies like FSGS, minimal change, lupus, diabetic nephropathy, and so on. This example is one of my favorite. It's an older image we have done very early in our developmental state at the Nipoca time. And you see, this is a part of a glomerulus of a biopsy. And for example, if you would analyze as a pathologist this region, you would say here in green, the filtration slit, in red, you have the synaptopodin indicating the food processes. Oh, this kidney is healthy. However, if you look a little bit on the right-hand side, and you see it here in a higher magnification, then you see that there is severely affected the morphology of the podocyte food processes. Now, we have had later on the problem with an animal study of knockout animals for actin-binding protein, which is relevant in podocytes. It's called pallidin. And so we have started at the beginning in a classical way with electron microscopic illumination. And very often we have seen healthy situation, like you see it here on the left-hand side, you see the podocyte food processes. Then we have had some sections where we have had such like a change of the food process morphology, but it was not really clear. Therefore, we thought, okay, is it possible to stain the food processes not only from human material, also from animal models with this technology, and to analyze it in a quantitative way. You see here, I think it's a fantastic view on podocyte, and here's the filtration slit and the interdigitation of the food processes. And we have done this indeed. So as an example, you see it here with a scanning electron microscopic image, example here with a broadening area. However, now it was possible really to quantify the situation in the pallidin control animals, as well as in the knockout animals, and also in a time-dependent way. So you can use this for all basic and preclinical research, and you are not only looking to one glomerulus. Normally we take 20 glomeruli with a thickness of 3 micrometer for the analysis. So in the past, we have had the luck, great cooperation with pharmaceutical companies to demonstrate the power of our technology. You see here the control situation. In the middle, you see the affected after injection of interferon, and in addition with a specific compound the Vertex company have looked for. And you see here the clear rescue of the disease after the addition of the compound 3 in this example. We have had also the luck to work with Gupra together in the nephroprotect effect of semaglutide, and you see it again here under different treatment conditions, semaglutide or semaglutide and lisinopril in an animal model of UNX renin, AAV mice. You see the quantification and the significant increase of the FSD, meaning the benefit of the podocyte morphology by the treatment. Now, in the past, we have now, I think, more than 20 or 22 publications showing that this technology works very well for different models, disease models like NTS, for example, for FSGS, or PAN, also for FSGS, Docker salt, for hypertensive-induced disease. And you can see here, we can also use it for rat, human, mouse, non-human primates, pigs, and also for organoids. You can see here an example, an organoid, well-performed food processes, and you measure it in a similar way as you have done it for the in vivo situation. You see it here on the right-hand side. So I would like to show you some examples from human biopsy studies. So in a great cooperation with Evotech company, we have analyzed 69 biopsies from the nurture patient cohort in a blinded way, of course, always in a blinded way. And what we have found after sending the FSD measurement back, the values back to the company, and the bioinformatic have analyzed these with the patient data, we have seen that we can categorize only by FSD the different glomerulopathies. So, for example, this is FSGS. You see that mostly the FSD is very similar. This is one patient, this is one patient, and so on. This is typical for membranous, minimal change disease, sorry, this is membranous. And you see the difference if you're looking for IgAN or hypertensive chronic kidney disease. So meaning we can identify, and so we have also measured it from primary and secondary FSGS. We find the difference between primary and secondary FSGS only by measuring the FSD. So we also included our technology into a clinical study, and this was in a cooperation with Elox Pharmaceuticals, and we looked for 3 Alport syndrome patients before and after treatment. And this is a classical view you get from 2 different pathology departments. It's not quantitative. Before the treatment with the drug ALX-02, it was widespread, the situation, the food process effacement, and then it was only segmental. Here in 2 examples, there was a measurement on the EM slides to have some quantification. And during 1 week, it was able for us to measure the difference between the 3 patients before and after treatment in an unbiased and really clear way. And you see patient 3 has had the most beneficial effect from the treatment from 160%. Now, there is also the possibility to include additional protein stainings. So for example, if we would like to know something about a protein of interest with the EM, it's highly sophisticated and a big challenge to find the immunogold for the specific proteins. And here on the right-hand side, as I have shown you, it is possible to make double or triple staining. And you can also look for other components of the filtration barrier, such as here's the endothelial cells, it's the capillary looms, the GBM, and the podocyte foot processes here on this triple staining. For the GBM staining in combination with the slit membrane, but you can also stain mesangial cells and to make later on an quantification. Now, I think this is the next generation of analysis in nephrology, and I hope I could convince you that this is really the future in determination changes of the podocyte foot processes. So at the end, I would like to show you some data about a zebrafish model we have established at University Medicine in Greifswald. And we use the zebrafish as a model for drug screening for FSGS. As you know, mostly FSGS has no healing drugs available. And if you are using mice or rats, it's really a very long story until you get the first results from a drug, whether it's working or not, and drug screening is not possible. So we have established a zebrafish strain, which is fluorescence. And fluorescence in all the vessels, as you can see here in green. And this protein, it's a vitamin D binding protein, which is coupled with a green fluorescence protein, cannot pass the filtration barrier, only pass the filtration barrier if there's an effacement of the podocyte foot processes. Here in the middle, you see this is a small kidney of the zebrafish larvae. It's called pronephros. It's only one glomerulus, and it's very similar to the glomerulus of human, rats, and mice. So what happened if you make a FSGS, for example, and you see it on the bottom, the movie, you lose the fluorescence, one from the capillary, because vitamin D binding protein is secreted, or pass the filtration barrier, which is leaky, as well as you lose the podocytes, which are labeled by M. Cherry. So these podocytes can be observed by multiphoton microscopy, as we have done it several times. You see here in a living animal, the podocyte cell bodies and major processes, but you cannot see the foot processes. This is, as I would like to mention again, below the optical resolution of a normal laser scanning microscope. No, what we have done is that we have used this zebrafish not only to visualize podocyte and whether the filtration barrier is leaky, but also to induce FSGS. And this is done by a specific technology. So the podocyte express not only M-cherry, but also the bacterial nitroreductase, NTR. And the NTR is normally inside of the podocytes and has no effect. However, if you put MTZ, or later we have used NFP, to the tank water, then this MTZ can pass the membrane and will be transferred from the nitroreductase into a toxic substance. And this toxic substance induces a detachment of the podocyte of the podocytes. You see it here on the left-hand side. You have all the podocytes covering the capillaries. On the right-hand side, we have strongly used the MTZ. You have a nearly complete depletion of this podocytes from the GBM here in green. So we have established this and looked, and all typical hallmarks are visible in this zebrafish larvae after a few days. And you can also study the morphology here with EM. In yellow, you see the GBM, and you see an effacement. We have matrix accumulation. We have podocyte detachment and activation of the parietal epithelial cells, which are signs for FSGS. So what we have then done is in a 96 well plate, we have put always one larvae and treated them with MTZ and MTZ together with specific drugs of a drug library. Then we measured the fluorescence, and each animal who has had still the fluorescence in the vessels or in the pronephros, in the kidney of the zebrafish, is a positive drug. So we measured this in an automatic way. So you see it here, and you see the zebrafish larvae is living. And what we have seen after the screening of an epigenetic library with 138 compounds, in cooperation with the Fraunhofer Institute in Hamburg. We have had 3 months of time for this. We have identified then a specific drug, and it's named belinostat. And we have had 3 different readouts. 2 were the fluorescence readout, as well as edema formation, which is shown here. If the larvae are highly affected, then you have a severe pericardial edema, as well as around here of the eyes. And then you collect and count how many animals react. And what you can see here, if the empty set, you have really a lot of animals with different stages of edema formation. And if you use, for example, a specific drug labeled here with 171, you have a strong reduction. So we have done this measurement under different conditions: co-treatment, continuous treatment, curative treatment. You can do everything you like. And we have found out that belinostat is one of our top hits. We have found more than 10 hits, and we are now working on the validation process. And then you can also include the histology from the same larvae you have looked in your microscope, in your screening microscope. And what we have found is, so here you have the healthy situation, you have podocin in M. cherry expressed, you see it after staining with an antibody. So if you have FSGS generation, then you lose your fluorescence specific for the filtration slit. However, if you use your drug, then you have a recovery, and also the morphology of the glomerulus, this is healthy and this is diseased, change after the treatment. Okay, now I'm at the end of my talk, and I hope that you have had, a little bit something interesting for you found in this talk. And at the end, I would like to mention all my crew members, cooperation partners. Without it wouldn't be possible to work in such an amazing way together. I'm really very happy to have such a strong group at the Department of Anatomy and Cell Biology, as well as the members of my startup, Nipoca. And I would like to thank you for your interest. And I'm really sorry that you have not the opportunity to ask questions. However, if you have some question, please write me an email. Thank you very much, and goodbye.